Hi everyone, in this lecture we're going to talk about genetic mapping in eukaryotes. Up to this point we've talked about independent assortment, and we've seen what happens when genes are on different chromosomes and how they sort independently in meiosis. We've used the chi-square test to show independent assortment, but sometimes the chi-square test might fail because the genes are not sorting independently. In that case the genes are linked. So let's see how this works. Question number 1. For a test cross of a dihybrid, which means a double heterozygous individual, the percentage of recombinants observed in the progeny will not normally exceed what? In a cross between two individuals whose genotypes are big A, big B, where big A is dominant to little a and big B is dominant to little b, the progeny are observed to show the following phenotypes: 1800 big A, big B, 600 big A, little b, 600 little a, big B, and 200 little a, little b. This data indicates that genes A and B are what? And 3, consider the following arrangement of alleles on two homologous chromosomes. Crossing over between these two genes during meiosis would result in which of the following recombinants? Genetic linkage is the case when independent assortment fails, when genes are not on different chromosomes. In fact, when genes can be on the same chromosome. The discoverer of linkage is Thomas Hunt Morgan, one of the founders of modern genetics. He established Drosophila as the model system. He discovered sex linkage in the early 1900s, and he established that genes are carried on chromosomes, so a pioneer in the field. He would cross fruit flies that had double mutations, for example, a fruit fly that had the purple eye mutation, and a fruit fly that also had the vestigial wing mutation, which is a really small, unformed properly wing. So these flies don't normally fly. They're kind of an oxymoron. So he crossed these double mutant flies with wild type. We're using little p, the symbol little p, for purple, and we're using little v for vestigial, and big P for wild type, or red eyes, and big V for wild type wings. Crossing these two flies together gives all wild type offspring: big P, big V, little v. Test crossing these with purple eye vestigial wing flies resulted in some strange results, not the normal results that we saw with independent assortment. The observed numbers are shown here in this column in the middle. The number of wild types exceeded what you would expect to find from an independent assortment. And the same with the purple vestigial double mutation, exceeded the number you would expect. However, the single mutants, the normal eyes but vestigial wings, was much reduced compared to what you would expect. And same goes for the purple and normal wings. So what's going on? Why are these numbers not matching to expected numbers? We expect a 1-to-1-to-1 ratio in the phenotypes. An odd result was when Morgan crossed flies that had vestigial wings but normal eyes with flies that had purple eyes but normal wings. The genotypes are different, and yet the F1 all would still be the same genotype as the previous cross: big P, big V, little v. And when test crossing should give the same results as the previous cross. However, very different. Now, the single mutations, vestigial or purple, show greatly enhanced numbers compared to what we expected. And the wild type was greatly reduced, as was the double mutant. What the heck is going on? He attributed this to, well, first of all, he noticed that the parental phenotypes were always overrepresented. In the first case, the wild type was apparent, and the wild type turned out to be overrepresented in the test cross. So what's going on? So his idea that some of these traits are linked, some of these genes are linked on the same chromosome. Remember, he discovered the idea that genes are on chromosomes. He had this idea that genes are linked along chromosomes in sort of a linear fashion. Genes that would be on the same chromosome then would not be independently sorting. not giving you the 1-to-1 to 1-to-1 ratios. Here's what he envisioned. Chromosome, the orange chromosome here would have the big A and big B alleles, and they would make gametes that would only have big A, big B. The chromosome here, little a, little b, would be present, and again would make gametes with little a, little b. We call these parental gametes. And these are the ones that were overrepresented in his crosses. Here's how we diagram this cross. I'm introducing a little bit of new terminology, new symbols here, where the two alleles are now linked on by the same line, which represents a chromosome. Homologous chromosome number 1 would be on the top and chromosome number 2 on the bottom. So a double mutant, purple eye, and vestigial wing fly would have this genotype, little p, little p, little v, little v. But the linkage would be between the purple eye gene and the vestigial wing gene. Crossing that fly with a wild-type fly on the right gives the F1, which are all wild-type. But notice that one of the wild-type chromosomes, the homologous pair, is still linked here in the F1. Test crossing then gives you those parental gametes that we saw and parental offspring. So one parent, or one F1, big P, big V, came from the original wild-type parent, and little p, little v came from the original purple eye vestigial wing parent. So half of the offspring are heterozygous, and half of the offspring are homozygous. And there's no mixing of the purple eye and vestigial traits. And that's because they're not produced. But if you remember from the tables that I showed you, the other phenotypes were showing up, but just at a really reduced number. So they must show up somehow. How do you get these numbers? So one mutation, the vestigial, but the normal eyes are showing up at much reduced numbers. So what's going on there? Well, this is where a concept called recombination comes in. We've talked about recombination, how homologous chromosomes recombine during prophase and metaphase of meiosis I. And that's exactly what Thomas Hunt Morgan discovered. The non-crossover or parental gametes are shown here on top. I'm sorry, shown here on the top, big A, big B, and in the bottom, little a, little b. But occasionally, but recombination can happen, and occasionally we'll mix up the genes. This is usually infrequent, and so a low number of recombinants are seen. But when recombination does happen, as shown here by the crossing over, then crossover gametes are formed, and the mixing up of the big A with little b and little a with big B. How does this look? Here's that cross from before with the double mutant crossed with a wild type. The F1 are heterozygous, and when you test cross them, if a recombination event happens, a crossing over, then the purple eye will cross over and be linked to the wild type wing, and the wild type eye will cross over and link to the vestigial wing. You'll end up with crossover gametes. When those crossover gametes are fertilized by a gamete from the test cross, then you end up with the recombinant offspring, vestigial wings and purple eyes only. The number of recombinants that you can get can never exceed 50%. When you look at a cross, a complete independent assortment, 1-to-1 to 1-to-1, 50% of them are recombinant and 50% are parental. So recombination can never exceed 50%. The parent phenotypes will always be at most 50% as well.